So thank you very much for the kind introduction, and also I'm very pleased to be invited to this very interesting conference. So I will actually talk today a bit about how we inherit mitochondrial DNA and what is going on with the maternal inheritance. So first I will just give you a little bit of a crash course on mammalian mitochondria. So as you know, the mitochondrial DNA is not naked. It's situated in a protein-coated structure called a nucleoid, and it's completely protein-coated and packaged as single copies. Also, it's now quite clear from human genetics, experimental work in the mouse and other types of experiments that the major sources of mitochondrial DNA replication are not reactive oxygen species, it's replication errors. So the DNA polymerase makes errors as it replicates mitochondrial DNA. And if you isolate mitochondria, you grind them up, you find around 1,200 proteins. And the mitochondrial DNA only encodes 1% of all of those proteins, so that's like 13 proteins. And to express these 13 proteins, as Massimo told you, you need to import around 200 proteins involved in transcription, replication, the mitochondrial ribosome. So it's quite a complex system. And as most of you know, the OXPHOS system consists of around 90 subunits, but without mitochondrial DNA, the OXPHOS system completely collapses. So the mitochondrial DNA subunits are few, but they are absolutely essential for oxidative phosphorylation. And another weird thing about mitochondrial DNA is, of course, the heteroplasmy that you can have in a cell or in an organism, in a human being, mixture between normal and mutated mitochondrial DNA. So most of our cells in the body have a nucleus, right, except red blood cells and platelets. So what is the origin of the eukaryotic and nucleated cell? Well, most evolutionary biologists nowadays believe that the eukaryotic cell was formed 2 billion years ago by a fusion event between an alpha protobacterium and an archaeon, where the archaeon became the nucleus and the alpha protobacterium the mitochondrion. And most of the genome of this bacterium then moved on was lost, and some genes moved to the nucleus. So this situation that we all live with, I guess, also created a conflict between two genomes, which is one explanation, I guess, for all of the pathology we see associated with mitochondrial DNA. And if you want to read more about this, you can look in papers, for instance, by T. Zetterman. So one person who has been thinking a lot about the transmission of mitochondrial DNA and organelle genomes was William Berkey, and he wrote almost 50 years ago, after having looked at many different situations, that there were usually the organelle genomes or the chloroplast or mitochondrial DNA was only inherited from one parent. And he also stated that this will then create a population genetic system, basically, not the Mendelian system. And some years later, which was still some time ago, he also concluded that there is no single mechanism for uniparental inheritance, and also he made the observation that there is no recombination when the genomes are inherited. So why is this important? Well, so I should say also it's very clear from studies of mammalian mtDNA already in 1974 was presented that in mammals the mitochondrial DNA is maternally inherited, and Doug Wallace also showed this for human mtDNA in 1980. So in the early 2000s was a lot of reports about the recombination of mitochondrial DNA in the germline. And we then did an experiment. So Eric Schubridge had generated heteroplasmic mice, which had a mixture of 2 different mtDNA genotypes. And those were generated and brought by Brendan Battersby to Finland. And these mice had been kept for 50 generations with 2 mtDNA molecules side by side. So when we did PCR on this, we found plenty of recombinant, but this was all artifacts because the PCR, in the PCR reaction, the enzyme can jump from one template to another. So this was before the era of long-read sequencing. So what we did, we cloned these into bacteriophages and sequenced them, and basically we didn't find any recombinant genome. So this sort of germline recombination data from us and many others argue that it doesn't exist. So why is this important? So the red dot here is a mutation in the nuclear DNA or in the mitochondrial DNA. And in the nuclear DNA, you have recombination in the germline, and then you can actually get the clean copy where the mutations is on one copy. But this is not possible in the mitochondrial DNA. And this is actually an asexual transmission of a genome. And a person who thought a lot about this was Hermann Müller, who already in 1964 wrote that this sort of asexual transmission will lead to a mutational meltdown. So, and this we call the Müller-Ratchet mechanism. So basically, if you don't have a sexual transmission of a genome, you can only accumulate mutations. And here is Müller, actually. He's an interesting guy. He got a Nobel Prize for X-ray-induced mutagenesis in 1946. And he also tried to do genetics under Stalin. He was American, but he moved to the Soviet Union and tried to do genetics. But it didn't work so well because Stalin supported Lysenko, right? So he had to go back to the United States, but still a great thinker. So why don't we get this mutational meltdown? Well, I think there are 2 major mechanisms counteracting this. One is the genetic bottleneck for mitochondrial DNA transmission, and the other one is purifying selection. So this bottleneck for transmission was discovered already by Hauswirth in the early 1980s. So he basically genotyped mtDNA in Holstein cows and showed that the genotype could change in just a few generations. And what is the explanation for this? Well, of the pool of mitochondrial DNA variants in the mother, only a subset are transmitted to the offspring. So basically, every time an oocyte is produced, just a subset of the mtDNA molecules in the mother are selected to populate the oocyte. So here is a human pedigree that I actually had in my thesis, which I think also illustrates this context. So the person with the thick square hair has the MERF syndrome and high levels of the MERF mutation. And as you all know, we don't find individuals with 100% of the MERF mutation. So it's clearly a selection going on. And you can also follow this mutation in the maternal line. So the original pedigree is much larger. This is just part of the pedigree. And to the left, you can see a woman with 33%, right? And she actually gets a daughter who has 0%. So this actually cleanses the mutation. The mutation disappears in the offspring to this woman. So clearly, the bottleneck can get rid of mtDNA mutations. And the way we think the bottleneck works is basically, so this is now the cycle or life cycle of mouse mitochondrial DNA. So after fertilization, the mouse oocyte or mammalian oocyte contains several 100,000 copies of mitochondrial DNA. And these are just distributed in the early embryo, and mitochondrial DNA replication doesn't start until after implantation. And after implantation, the primordial germ cells are set aside, and these cells will then give rise to the future oocytes. And at this stage, there is a very low level of mtDNA. So some years ago, we made a mitochondrial DNA mutator mouse. So the DNA polymerase that controls the replication of mitochondrial DNA is encoded in the nucleus and imported to the mitochondria. And we just made a subtle mutation so that this polymerase would get sloppy and create a lot of mutations. And this actually created quite a bit of attention, and we even got the cover page of Nature, and it's actually my most cited paper. So these mice got a lot of premature aging syndromes. So this was sort of experimental evidence that mitochondrial DNA mutations can induce aging-like symptoms. So concerning inheritance, I want to make a point here. So to get the mutator mouse, which is with this red dot here, you have to mate 2 heterozygous mice. And you can see the heterozygous mother to the mtDNA mutator mouse. They create low levels of mtDNA mutations that are inherited. So this mutator mouse to the bottom right here has both a lot of somatic mutations generated by the sloppy polymerase, but also inherits mtDNA mutations from the mother. And we then decided, can we eliminate these inherited mutations? And we did this by, instead of a heterozygous mutated female, we used a heterozygous knockout female. So then the red circle here is this mtDNA mutator mouse that now have no inherited mutations. And actually, this is just the lifespan curve, and you can see the red curve here. So normally, the mtDNA mutator mice live for 40 to 50 weeks before they get sick because of all of these mutations. And if you eliminate the maternal contribution, the mice can live 10 weeks longer. So clearly, the inherited mutation are important and provides a basis for somatic mutagenesis. So if you inherit a lot of mutation and have somatic mutagenesis on top of that, it will be worse. And this is also relevant for humans. There are many papers from Patrick Chinner and others showing that heteroplasmy in humans is universal, and we quite often inherit mtDNA mutations at low levels. So what about germline purifying selection of mitochondrial DNA? So of course, all of the human data are observational, and we some years ago did quite a simple experiment. So we took this mutator mouse and bred it for several generations. So the mtDNA mutator mouse, here we sequenced the cytochrome B gene in different tissues, and you can see that all codon positions of protein coding genes are affected to the same extent. So there is like a random mutagenesis going on. But after a number of generations, in this case 6 generations, we were able to create lines of mice which have high levels of just a few mutations. And then we also bred away the mutated polymerase. So these mtDNA mutations were now maintained by a wild-type mitochondrial DNA polymerase, but because of the repeated passages through the bottleneck, they randomly accumulated to high levels. And then we sequenced these, and we saw a very strong selection against mutations in the first and second codon position. So this purifying selection is quite rapid and quite strong in protein coding genes. So this is, of course, also relevant for humans, and here is just one example from Patrick Chinnery, where they looked at human embryos and saw this depletion of mitochondrial DNA, these low levels of mitochondrial DNA in primordial germ cells, and also evidence for purifying selection. So by breeding the mutated mouse, we also generated this tRNA Alla mutant mouse. So this was done by Joanna Kaupila and Jim Stewart, who was then in my lab. And these mice have this pathogenic tRNA mutation, which has been used quite a lot by different groups. So basically what this mutation does, it makes the tRNA unstable. So if you have high mutation levels on the x-axis, you can see then that the levels of the tRNA on the y-axis go down. And if you do isolate heart mitochondria and look at the inorganello translation, you can see that with high mutation levels, the translation go down. So these animals get a mild cardiomyopathy with a mild increase of the heart mass. And they also get cytochrome C oxidase deficiency in colonic crypts. But yeah, so when we compared the levels of mutated mtDNA in ear clips, which is the x-axis, and different organs, in this case heart and colon, at the age of 20 weeks, you can see there is a fairly good correlation. But as the mice grow older, 50 weeks or 75 weeks, you can see the red dots, which are the mutation levels in colonic tissue, which is cells that can divide. There is purifying selection, because the initial levels that you find in the ear clip are then much lower in the colonic tissue. And when we did this in the presence of decreased mitochondrial DNA copy number, this purifying selection was even more marked if you compare the situation of the graphs to the right, to the left and to the right. So with this mouse, we can breed them and get females that have around 80% of the mutation. And if we breed these females, so the mutation levels in the mother is on the x-axis, and the mutation levels in the offspring on the y-axis. You can see if the mother has very high levels, she can only get offspring with lower levels. But if you, for instance, look, if the mother has 40%, you can see that the offspring can get both higher and lower levels. So here is clearly a threshold for the transmission and the purifying selection going on. So to experimentally investigate what is going on, we decided to use these mice and see how impaired autophagy would affect the transmission. And this project was done by a postdoc in my lab, Laura Kremer. She's now back in Göttingen and starting her own group. And we also collaborate with Lyuba Boshilova, who is a statistician, and Patrick Kramer. Sorry, not Kramer, Chinnery, sorry. So here is now this mutation level. So basically what we have on the y-axis here is the normalized heteroplasmic shift. So you cannot yet get mothers where every mother have the same levels of mutation. So we just normalize the difference in mutation levels between the offspring and the mother. And the red arrow indicates if there is no purifying selection, the mean would be on this dotted line. But you can see in these control groups, there is always lower. So there is clearly a purifying selection against this mutation in the germline of these mice. So then we looked at different mutations that supposedly affect autophagy. Parkin doesn't do anything. We also had generated in my lab knockout mice for BCL2L13, which is the mammalian homologue of yeast, ATG32. And you can see in this mutant basically abolishes the purifying selection. And when we have combined with knockouts of ALK1, ALK2, which is the initiating kinases for autophagy, it's called ATG1 in the yeast, right? We also saw an effect. So autophagy seems to play a role for the transmission of this mtDNA mutation. So what about Parkin? Well, I think this idea that Parkin is involved in mitophagy is really poorly supported by in vivo evidence, and there's quite a bit of literature on this, and here is just a recent paper from us that we published in 2024, where we basically aged the Parkin knockout mouse, and they were quite normal, no effects on bioenergetics in skeletal muscle. Also, we didn't find any effect on this pro-inflammatory response. And we also disrupted Parkin in dopamine neurons, because there has been this idea that you don't see much in the mouse because there is embryonic compensation, but nothing happened. And why we finally decided to publish this is that we also did a muscle biopsy on a Parkin deficient patient, a lady who got sick in Parkinson's disease in her 20s, and we did this muscle biopsy when she was 50 years old, so 50 years without Parkin, and the respiratory chain function in skeletal muscle was normal. We have quite a big experience with measuring this, as the clinic has done over 1,000 muscle biopsies. So another interesting mutant we started working on for the wrong reasons is this FBXL4 deficiency, where there are many patients described with this mutation, autosomal recessive mutation in FBXL4. And why I got interested in this is because I thought it had something to do with mitochondrial DNA replication, because all of these patients have low levels of mtDNA. But what we actually found when we did a knockout mouse and also studied patient fibroblasts is that these patients have a deficiency of mitochondria and an increased levels of lysosomes. So there seems to be an increased autophagic turnover of mitochondria in these patients. And a few years after we published this initial report, came two nice papers from other groups in EMBO reports, which basically showed what this FBXL4 is doing. So it's actually part of SCIP1, CAL1 complex that ubiquitinates receptors on the surface of mitochondria. And if this is not occurring, you then get suppression and high levels of these receptors on mitochondria, and they are turned over by mitophagy. And actually, what was also shown by these groups, and you can see here the red line is the survival of the homozygous FBXL4 knockout mice. So we only got 5% instead of 25% viable mice when we mated the heterozygotes. So this is actually the same frequency as these authors got. So the red line, they have like 20% of the expected fraction. And when they combine this with knockout of mitophagy receptors like NIX1 or BNP3, they get more or almost normal proportions of viable mice. So deletion of NIX1 and BNP3 can suppress this phenotype. And this is interesting because there has been quite a bit of work in the fruit fly on the inheritance of mitochondrial DNA. where work has shown that the fragmentation of the mitochondrial DNA network is important. And also in genetic screens, there has been an identification that the ATG1, which is done in mammals, called ALK1, ALK2, and the BNP3, which in the mammals have two variants, BNP3 and BNP3L, also called NICs. So these molecules are all important for germline mitophagy in the fruit fly. So we recently did quite a massive experiment to investigate whether the bottleneck and autophagy interact during purifying selection of mammalian mitochondrial DNA. And this was again done by my postdoc, Laura Kremer, and we collaborated here with the group of Patrick Chinnery. So this is just a bit of a complicated breeding strategy. But basically what we did, we made sure that we generated mtDNA mutated mice by the strategy I previously described, so that we have mtDNA mutated mice that don't inherit mtDNA mutations from their mother. And then we bred them for 3 generations to allow the mutations to segregate. And we did a lot of high-depth sequencing of the mitochondrial DNA. And to study the impact of modifying factors, we combined this with increase of mtDNA copy number by overexpression of TFAM or a decrease by heterozygous knockout. And we also manipulated autophagy by knocking out BCL2, L13, and ALK2. So all of this breeding is a bit complicated, and if anyone wants to know more, just talk to me afterwards. So what Laura did, she sequenced 161 mice from the different crosses, found more than 3,000 mutations, and here is just a circus plot where she plots all of these mutations around the mitochondrial DNA. And you can see the variance per base pair, if you look at synonymous mutations, are higher than non-synonymous, so there is clearly purifying selection going on, which is reminiscent of what we see in humans. And also we saw less mutations in the cytochrome C oxidase 1 gene and more in the ATP6 and 8 genes, which also is consistent with the pattern seen in humans. So what happens? So this is now the heteroplasmic fraction, basically the level of heteroplasmy. So if we decrease mtDNA copy number, you can see that the heteroplasmy levels get higher. And if we decrease autophagy, the heteroplasmy levels get lower. And with BCL12-13, we had a bit of an effect. So what about mutations, the number of variants? So if you have a tighter bottleneck, so you have a reduction in mtDNA copy number, you get, as I showed in the previous slide, a higher variation in the levels of heteroplasmy, but a stronger purifying selection. And if you have a wider bottleneck, more mtDNA copy number, you get a decrease of purifying selection. And with the knockout of ALK1, also you get more mutation, decrease the purifying selection. So basically, I think these experimental data are quite clear, showing that if you have a tighter bottleneck, you have a higher heteroplasmic fraction, stronger purifying selection, decreased mutational burden. If you have a wider bottleneck, we couldn't see any effect on the heteroplasmic fraction in our experiments, which may be due to technical reasons. And the purifying selection was then weaker, and we had an increased mutational burden. So if we modify autophagy, we have an impairment of the general autophagy, slower mitochondrial turnover, lower heteroplasmic fraction, and a weaker purifying selection, more mutations. And with the BCL1213, we think that this is mainly involved in pathogenic variants with high heteroplasmy, and therefore we don't see so much of an effect on the total mutational burden. So before I end, I would just like to make a few points here. So first of all, there are many mitophagy receptors, and these are likely, at least several of these are likely to be important for purifying selection in the germline. We have studied BCL1213 in the fly. EPNIP3 is shown to be important. Patrick Schinner, in a recent paper, showed that there seems to be a down-regulation of the proteasome if you have high heteroplasmy levels, and if you suppress the ubiquitinase enzyme, actually you get more ubiquitin on the mitochondria and more purifying selection. And something quite interesting that is appearing on the horizon is also this phosphatase, which seems to be regulating mitophagy and interacting with this FBXL4 complex. And finally, this is just a paper out showing the quite big complexity in the way these autophagy receptors are recognized by different protein complexes on the phagosome. So conclusions from our work is basically that mitophagy is very important for purifying selection of mitochondrial DNA mutation in the maternal germline. The bottleneck for maternal transmission is directly linked to purifying selection, so copy number plays a clear role. And mitophagy and the proteasome, I think, will act in concert, so they will not act independently, but their functions are interlinked in purifying selection. And I think this also opens up the future drug development to actually manipulate this system to prevent or decrease the transmission of mutant mitochondrial DNA. So I will end here, and most of the work I've been talking about today was done by Laura Kremer when she was a postdoc in my lab. And there are also other members of my group depicted here. And this last part with this massive mouse breeding was done in collaboration with Patrick Schinner in Cambridge, and Sue Golder and Tom Barton Owen. So I stop here. Thank you very much.